It now falls on me to introduce Matthew Pickering. He's Professor of Rheumatology at Imperial College, and he's Mr. C3 Glomerulopathy. He runs a complement-mediated disease centre in Imperial, and he's also a consultant rheumatologist. Matthew.
So thanks very much. I didn't know about YouTube, actually. I didn't go to makeup or anything. I mean, so this is, so the last time I talked on complement, it was put on YouTube, actually, and I think we're up to maybe 10 hits or so now. So my children find it very amusing. Okay, so complement. Well, complement's a very interesting subject from my point of view, best discussed after coffee, so it's a perfect timing. So let's think about what this means. And I'm informed that what I should do at the end is give you a little test, okay? And according to how you perform, then you get a lunch voucher, and we can break for lunch, okay? So I've come up from London. I work, as Kevin said, in, let me just find the mouse, the pointer here. Yeah, here we go. Oh, that's not very bright, okay. In Imperial College, and we run a centre where we're interested in complement and how complement causes disease. And we work in the laboratory. We use test tubes. We use animal models to try and understand kidney disease. And of course, we see patients. And we see patients also in the same area, in Imperial College in London, in the Hammersmith. And I run that with my colleagues who are here today, Dr. Tom Cairns and Nicola McKenna in the centre here. And we'd love to interact and discuss with you any questions towards the end about our service. Okay, so what is complement? Well, if you look up a definition, it's a thing that contributes extra features to something else in such a way as to improve or emphasize its quality. And that's where it really derives its name when we think about its function. So I'll explain more about that shortly. So there's an example. Local ales provide the perfect complement to fine food, okay? And the important concept here is to bear in mind that the food remains fine even without the local ales, okay? So in studying complement, we know if we take away complement, okay, and complement's role is to fight infection, it's not a total disaster. We're still able to fight infection, just not perhaps as well as we would be if we had an intact complement system. And that's really important when you think about drugs that switch off complement, okay? Now, here's the sort of medical school definition, a group of proteins present in blood, plasma, tissue fluid, which combine with, it goes on, and you lose the will to live, don't you, with that sort of definition. That's not very helpful at all, so we can do a lot better. And I think whenever I'm trying to explain things and when I understand things, I go right back to the beginning and say, well, who discovered complement? You know, where did it come from? Because by understanding that, we really get a grasp as to what's going on. So, now, this is a picture of a complementologist. We don't look so serious these days. This is Jules Bordet, and this is back in 1895, okay? So this was a time when infection was the key problem in human health, all right? And he was interested in how bacteria are destroyed and worked out very cleverly that you required something called an immune body. We call it antibody now, and that's shown there. You can see antibodies. But also something in the blood which you could destroy just by heating the blood up, which is complement. Okay? Now, Jules Bordet called it alexin, okay, because it comes from the meaning to ward off the bacteria. And that's where alexin gets its name. That's interesting, isn't it? But then enter another really Jedi Knight of immunology, Paul Ehrlich, who preferred the name complement and switched this term to complement. And he writes here, look, an adamant in blood, which together with this immune body, the antibodies, destroys the bacteria. Okay, so this is what complement is doing. And here's the first depiction of complement, okay, from Ehrlich's writing and laboratory notes. So we've got lunch in about an hour. We've got to get from 1895 to 2013. We can do this very, very quickly, and it's not a problem. And really, what's happened over that time is we've understood exactly what's in that little schematic I've shown you. Okay, so what are the pathways? What's complement made of? It's a protein network. And then more latterly, really from about the 1970s, we've understood what happens when complement goes wrong, and that's complement and disease. And then much more recently, we started to design drugs that can manipulate complements. And many of you would have heard of the drug eculizumab, and that targets the complement protein. Okay, so let's go through them. So what is complement? It's a protein network that combats pathogens. These are things that cause infection, as Kevin explained. It destroys pathogens in many ways using firepower. Now, I'm getting worried about scientists and military analogies, but I mean, maybe this is telling us something about ourselves. But we need to think about this. Firepower clearly aimed at destroying non-self bacteria. And it does it in many different ways. It can kill the bacteria directly, and this is fascinating, and I'll show you that in a moment. It can mark the target for destruction, or it can call for help. Okay, so this is essentially how complement works. And that gets rid of the bacteria. And a lot of early work on the system, as I said, this was a time when infection was a key problem in human health, was studying the relationship between complement and pathogens. And we've learned a huge amount about this protein network by studies of that sort. So let's look at that in a little bit more detail. What do I mean can kill the target directly? Well, it can generate these holes on the surfaces of bacteria. This has got a funny name. I'm going to minimize jargon, okay? So excuse me if I put things up, but I'll try and explain them. So this is called a membrane attack complex because the membrane is the outer part of the bacteria. So it's just a complex that attacks that membrane. And what happens is, if the complement produces this complex, which is a cylindrical protein structure that actually allows fluid to go through it, and it literally inserts into the bacteria's surface. Now the bacteria becomes leaky, okay, and explodes. All right, so that's the end of the bacteria. And that's one of the very potent aspects of the complement firepower. And we can see this if we use very strong microscopes. Look at that. Isn't that a beautiful picture? This is an electron micrograph, we call it. But these are holes on a surface caused by this protein complex. Complement can also coat the pathogen with, if you like, signals that say, this is non-self, eat me signals, all right? And the jargon here is opsonin. Okay, and this shows a bacteria coated with opsonins. These will be things like C3. We've heard of C3. And that then is, if you like, that's the sort of the sauce that goes on the food. That helps the cells, this is a depiction on the far right there of a human cell, a phagocyte, to clear that bacteria. And then, of course, complement can't work on its own. All right, we have to also alert other ways, other cells of the immune system to clear the bacteria, and it releases these substances that essentially produce a signal that says, calls for help. Okay, and the jargon there is an anaphylatoxin. And they're released around the bacteria, and as they diffuse away, they get less and less, and these cells will follow that gradient to find the bacteria. Okay? And then if we look at it in a little bit more detail, we say, well, let's get into some, because we need to understand how things like eculizumab work and what on earth the C3 is. If we look at the protein network and the firepower, we see that really it's predominantly coming from these two components in the protein network, C3 and C5. And you can see C3 is producing the eat me signals, C5 is producing some of these anaphylatoxins and the membrane attack complex. Okay, so from C3 and C5, we get the firepower. Now, of course, if you have that system, then it's an obvious thing, isn't it? Well, we've got to make sure that we recognize bacteria. We don't recognize our own body's tissues. So we have to ask the question, well, how does complement know that? How does it work? How do we distinguish between one of them and one of us? And this is the depicted schematically here. Well, it's only switched on when it senses pathogens. So how does it sense pathogens? Well, it can use these proteins called antibodies, which it's cheating, if you like. The antibody's doing the job. The antibody's recognizing the bacteria. It binds the bacteria, and that is a signal to the complement system to unleash the firepower. And that's got a name. That works through a pathway in that protein network complex that's called the classical pathway. It's one of the first things that complement biologists understood. You remember that at the first slide, it was something that enhanced the ability of these antibodies to kill bacteria. But that was not all. It was more complicated than that. I mean, bacteria look very different to us. Why? How do they look different to us on the surface? Well, they have different sugars than our own cells. And complement will spot those different sugars and trigger activation through a pathway called the lectin pathway. And that was discovered much more recently. Now, there's one part of the complement network which really can cause trouble and is very relevant to the kidney, and that is the part of the complement network that is always switched on. This is a very ancient way of fighting infection. This works on the principle that you attack everything, okay? Indiscriminate activity, and then it's up to the host to defend itself, okay? So you move the recognition, you turn it on its head. So, and this we refer to, this pathway as the alternative pathway. And again, this was elucidated in the sort of 1970s. Again, it's very relatively not that long ago when we think about things starting in 1895. So the alternative pathway will deposit this firepower on not just bacteria, but also on one of our own cells. Now, of course, that doesn't happen. So why doesn't it happen? Well, it doesn't happen because we've also developed in evolution proteins that switch off this alternative pathway. Okay, they don't switch it off when it's happening on bacteria, but they do switch it off when it's happening on our own cells. And we call these proteins complement regulators. Okay? And they do have strange names that relate to all sorts of things, like, you know, the way that scientific nomenclature is, people who discovered them, and so on. And the names really aren't important. It's just the concept. And let's look at some of them float in the bloodstream. There's factor H and factor I. And they will go everywhere where blood goes and protect surfaces. And others are on the cells. And they have, again, strange names, and some have more than one name. And some of these regulators, it's important to think where they come from. Many of these regulators are made in the liver. Okay, so that's relevant to the kidney. So if there's a problem in making one of these regulators, you don't solve that problem by putting a new kidney into somebody because it's coming from the liver. Okay, that's true for factor H, for example. And of course, when you have a normal complement regulators in the blood and on the cells, you don't get this indiscriminative. You're protected from damage from the alternative pathway. Okay, let's move now away from the biology of how it works and how it fights infection to thinking how it's relevant to disease and, of course, the kidney. So we can think of it really very simply, can't we? We can think, well, it has this firepower. It can either have too little firepower or it can have too much firepower. So what happens when you have too little firepower? Well, these were the first complement diseases described in around the 1970s. People were described who had deficiencies of C3 and C5. And what happens? Well, they're more likely to get infection. That's what you predict from understanding how complement works. And you can see here, here's the firepower. If we have no C5, we lose two components of it. If we have no C3, we lose the C3 part as well as the C5 part because it's upstream. So that's very relevant, isn't it, when we think about switching off complement with, say, medication, tablets. But the good news is, as I said earlier, if you have good functioning antibodies, then they can do the job most of the time to fight infection. So people with complement deficiencies tend to have a lot of infections when they're young, before they've developed all the antibodies that you develop as you age. But when you get into adulthood, you tend not to have so much in the way of problems with infection. Okay, it's still a concern, but it's not as much of a concern as we might have predicted. And then let's switch to the other way. Well, what about too much firepower? Diseases where there's a problem with protecting our own tissues from the firepower. Well, here you're going to predict maybe that could happen if we don't have those regulators working properly. Then we've got problems. And we can think of this balance of firepower as really being balanced by on switches and off switches. So if we've lost some of our off switches, we've lost some of those regulators, you remember that name, factor H, we've lost that regulator, then the firepower, okay, is going to get greater. We've got too many on switches. A good on switch for complement is infection. So what if we have not very good working regulator and we get an infection? That's going to disrupt that balance. Okay? We reduce the off switches, we increase the on switches, and we can get disease. And we know this applies to many diseases. And there's two in blue there. These have got terrifically difficult names, haven't they? Look at these. Atypical hemolytic uremic syndrome and damage to red cells in a condition called paroxysmal nocturnal hemoglobinuria. I mean, this is what doctors do. We put things into jargon, which confuses everybody, doesn't it? But what we're interested in today, of course, is MPGN and how this is going wrong in MPGN. So I would think of some components of MPGN, not all, it's never that simple, but some components of MPGN as really a disease where there's a problem with protecting our kidneys from this firepower. And we know there's not just MPGN, but there's different names. And all of this, look, we can encompass by C3 glomerulopathy. What you're looking at in the middle of the screen is that filtering unit of the kidney, and it's green because what we've done is we've put a green tag on C3. So what we can see in that kidney is lots of C3, okay, which is where the name C3 glomerulopathy comes from. There's all this C3. So why is it there? Well, let's think back to what we've learned. There's no bacteria. There's no signals for complement to put C3 in that kidney. There's no sugars on bacteria. There's no antibodies on bacteria. But how else could we get C3 without that? Well, of course, it's the pathway we know is indiscriminate, the alternative pathway. It's attacking everything. So is there something wrong with the switches? The C3 is not drawn to scale here. It's quite big, isn't it? Okay? C3 is a very tiny molecule. If you think of a golf ball, it's about 1/10,000,000th of the size of a golf ball, okay? Which is hard to visualize. But if we magnify the C3 up to the size of the golf ball then becomes the size of the moon. Okay? That's pretty staggering, isn't it? So let's look at a little bit more detail. Too much firepower in the kidney. What do we mean? Well, we've got this protein network. We have this pathway that's going to attack our cells indiscriminately. That's good for bacteria, bad for our host cells. Maybe we've got a problem. Let's choose factor H. It doesn't have to be factor H. That's just one of the switches. Let's choose that. We get more firepower. Now we're unleashing C3 and C5 and these products that we've talked about. And here's our kidney cells. They've got some switches on them. You can see just depicted schematically here. Now we get problems. We get inappropriate deposition of complement in that kidney. And that's what you saw. That's what the green picture showed you, that there's too much of one component of the firepower, the C3, in the kidney. And if you looked for C5, you would see that too. And if you looked for the membrane attack complex and these opsonins and these anaphylatoxins, you would find those too. So too much firepower in the kidney, a problem with the off switches, a problem with the on switches, giving you this disease. And remember, the on switches might be because we're born with very strong activating complement systems, or it may be that we've met something that activates complement, like throat infections. We know that infections like that make it switch on complement. And that's potentially a bad thing when we've got a problem with switching it off, of course. So then the final part of the story is complement drugs. And really, this can be done very briefly because we've learned a lot now about the firepower. We can look at this and say, here's our protein network, the firepower, C3, C5. Well, we do have a drug that will target specifically C5. And it's very specific for C5 because it's like it's an antibody. Kevin told you about antibodies that are like these homing missiles. This is going to inhibit C5 and C5 alone. It's eculizumab. It's marketed as Soliris, and it will switch off two components of this firepower. And depending on whether that's enough, that may be helpful in the disease or not. If C3 is causing a problem, it may not be enough. And at the moment, we know a lot about this drug, and Professor Goodship will talk a bit about this later on in these conditions, PNH and atypical HUS, but of course we want to know about its use in MPGN as well. So this is my last slide, and I finish with a concept that Paul Ehrlich introduced, which was a magic bullet. And this is a superb concept, and in some ways quite an intuitive one, which is, remember, Ehrlich is thinking about infection. They're not at the stage that they appreciate that complement damages self-tissue. Ehrlich is thinking, well, in order to destroy bacteria, we need to target that bacteria specifically, nothing else, and attach to that target something that will destroy the bacteria. That's the bullet, the magic bullet. It will achieve the objective with no collateral damage. And it's very interesting that now we are taking that concept further, and with eculizumab we have, if you like, a magic bullet that targets one component of the complement system and switches it off. And in development at the moment, and this is why the future is bright, are many, many ways of targeting the complement system. Okay, and these are not going to take decades to arrive. These are going to be arriving within years, and they include antibodies and other proteins that will switch off C3. Okay, so our motivation is to understand these diseases in as much detail as possible so that we're in a position to know how we would apply these new therapeutics. Okay, and I'll finish there and take any questions, and thank you for listening. Yes, just, yeah.
You're talking about low C3 and low C5 making people protein infection. Is that the same with low C4? And is it still
Yeah. Yes. Low C4, much less so, actually. So there is a relationship between which part of that network is missing or run out or deficient and infection. So C4 tends not to cause too much in the way of infection because you can still use C3 and C5 without going through C4.
Right, okay, then I just wanted...
Yeah.
I think that's us. We'll move on.
Thank you.